Hi, everyone. My name is Hope Avalone, and I'm the Senior Manager of Patient Education Programs here at the Lymphoma Research Foundation. It's my great pleasure to welcome you to our webinar on highlights from the 2024 American Society of Hematology Annual Meeting. During today's webinar, you'll hear from a lymphoma specialist about any T-cell updates, and you'll have an opportunity to ask questions. You can ask those questions at any time in the Q&A box at the bottom of your screen, and we'll address as many as we can following the presentation. Any question that is submitted through the Q&A box can be made anonymous, and as a reminder, this webinar is being recorded. And at the end of the program, you'll be prompted to complete a program evaluation. We do ask that you complete this evaluation because we incorporate your feedback into future programming. But before we get into the doctor's presentation, I did want to share a few words about our organization. The Lymphoma Research Foundation is the largest nonprofit organization that focuses specifically on lymphoma and chronic lymphocytic leukemia. Our mission is to realize the promise of science to eradicate lymphoma and to serve the community touched by this disease. And all of our work is led by our Scientific Advisory Board, which is a group of 45 leading lymphoma experts from around the country. And in addition to today's program, the Foundation has many other programs and services available for you. We offer both in-person and virtual programming so that you can continue learning throughout the year. So our in-person programs, or our lymphoma talks, our weekday dinner programs our weekday dinner programs. And in addition to our in-person program, we also have virtual programming, which include our Ask the Doctor About Lymphoma series, our Living with Lymphoma series, and our annual educational forum on lymphoma. And lastly, we also have our webinars like today's program. And if you'd like to view the dates for any of these upcoming programs, you can always visit lymphoma.org/programs. But for now, I'd like to specifically highlight our upcoming Living with Lymphoma program, which is a free virtual half-day program on Saturday, April 19th. And Living with Lymphoma provides detailed information on specific lymphoma subtypes and supportive care topics. So the program this time around will include a dedicated breakout session for T-cell lymphomas. And we are on a new platform this year called CVent. And on that platform, you'll be able to join community discussions and connect with other patients and care partners. So to view the agenda for this program and to register, you can visit lymphoma.org/livingwithlymphoma. And in addition to our programs, the Foundation offers a wide range of support services. So our helpline, which serves to complement our patient education programs, is a resource for both patients and care partners. The helpline has master's level trained staff who can provide you individualized information on any type of lymphoma, as well as information on treatment options, clinical trial navigation, and connections to resources like financial assistance. We also offer a peer support program called the Lymphoma Support Network. And through the Lymphoma Support Network, we can connect patients and care partners with others who have been through similar experiences for that emotional support. And we also offer comprehensive disease guides and fact sheets, which can be ordered or downloaded for free on our website, lymphoma.org. And also please look for us on YouTube, where we spotlight different lymphoma subtypes and treatment options. And finally, I'd also like to highlight our Spanish language resources for those of you who would find information in Spanish helpful. I'd also like to take a moment to thank our program supporters. Thanks to their generosity, we're able to host programs like today's webinar. For now, we have a wonderful program planned for you today, and I'm honored to introduce our speaker, Dr. Pierluigi Porcu. Dr. Porcu is a physician scientist focused on T and NK cell lymphomas. He is a professor at Thomas Jefferson University, where he's also the director of the Division of Hematologic Malignancies at Sydney Kimmel Medical College. He's participated in numerous patient education programs, and we're so pleased to have him back today. So Dr. Farku, thank you, and I'll now turn the talk over to you.
Thank you so much. Hope it's really great to be here, and it's been a great pleasure working with the foundation over the past many years on program like this and more. So I want to welcome everyone here. I'm going to start sharing my screen now. Hopefully, it will be okay. And see if is it visible?
Yes.
Terrific. And now I'm going to put it in presentation mode.
Looks good.
We are good. Okay, terrific. All right, so the text, it'll all go working good. So welcome everyone. Again, it's my pleasure to provide a brief update on the abstracts that were presented, abstract and oral presentations that were presented at the last annual meeting of ASH annual meeting in San Diego with a focus on T-cell lymphoma. I want to start by sharing my conflict of interest disclosures for everyone to see. And I'm going to start just with a very, very brief introduction on mature T cell and NK cell lymphomas or neoplasms, the way they're called now. I'm assuming that the vast majority, if not all, the people on this call either have or had T cell or NK cell lymphoma, or they have family members, or they have a special interest in this. And I think the first thing to say is that something that is well known to many people, and that is this is an incredibly diverse and heterogeneous group of cancers, blood cancers. That's part of the challenge that we've had over many years in identifying biomarkers of response and treatment for each one of these diseases. And one of the additional challenges is that most of them are quite aggressive. It's good to envision mature T cell and NK cell neoplasm in four broad categories that are shown here. Starting on the top left, you have the vast majority of the cases are so-called nodal T cell lymphomas. These include peripheral T-cell lymphoma not otherwise specified, anaplastic large cell lymphomas, ALK positive and ALK negative, and then this kind of evolving subset of lymphomas that are called follicle helper T-cell lymphomas that include angioimmunoblastic T-cell lymphoma, which is the most common one, but there are a number of other lymphomas with a similar phenotype that now falls into this family. There is some overlap between these entities, and so it's always important to have a very careful diagnostic workup done when you're diagnosed with one of these lymphomas. Another group at the left bottom is the cutaneous T-cell lymphomas, which historically have been sort of looked at and analyzed as a separate group or completely separate group. In reality, there are a number of activation signaling pathways and vulnerabilities in response to therapy in CTCL that are similar to the nodal peripheral T-cell lymphomas. And these represent about 20%, so about 2,500 cases, new cases per year. And then on the right, there is this group of leukemic or disseminated T-cell lymphomas and the extranodal lymphomas, which are very rare diseases, especially individually. So most physicians are not likely to encounter more than one or two of these cancers during their professional career. And some of them are aggressive, like adult T-cell leukemia lymphoma, Sézary syndrome, right there on the top, and others are fairly indolent, like LGL leukemia. So this is the spectrum of the diseases. The way we classify them changed slightly with the last 2022 classification. There are two classifications. One is called the International Consensus Classification, ICC. Another one is the WHO. There are kind of very small differences between them, particularly for T cell and NK cell malignancies. So I'm just going to show this one. This is something that we use all the time. And again, it goes through some of the it's almost like a laundry list of different entities. And there's no particular insight from this list as to what is driving these malignancies, which ones are curable or not. This is our resource that we go to with pathology to make sure that we actually classify the lymphoma in the best possible way. When we look at outcomes, these are data from the Swedish Lymphoma Registry, which is a national registry that captures more than 95% of all the lymphomas in Sweden, and therefore is fairly reliable as far as the accuracy and the completeness of the data that are being collected. And this includes only the nodal T-cell lymphoma. So the leukemic types and the CTCLs are not excluded. I'm sorry, nodal and extranodal, but the leukemic type and the CTCLs are excluded. So if you look at the top, these are the overall survival on the left and the progression-free survival on the right for the nodal types. And you can see that with the exception of ALK-positive anaplastic large cell lymphomas, which have a much better overall progression-free survival compared to all the other ones, are relatively unfavorable diseases with minimal long-term survival and certainly very minimal progression-free survival. If we look at the bottom, which includes the extranodal types, again, overall survival on the left and progression-free survival on the right, we look at the outcomes, and also they don't look very good, with the exception of the subcutaneous panniculitis type T-cell lymphoma, which has a better overall survival because there are a number of therapies that can be used at relapse, even in second, third, or fourth lines of therapy. So this is the kind of the context and the framework that we have to deal with when we are looking at what we need to do in T-cell lymphomas. In terms of the presentation that follows, I just want to remind the audience just to keep some things in mind. One, my selection of the abstracts is based on my personal assessment of the overall interest of the data presented in terms of the T-cell lymphoma world and also the impact. This doesn't mean that my view is the only one. There may be others who would present a slightly different combination of abstracts. The one thing that I'm not going to be able to do is I'm not going to be able to present in detail all the abstracts. Keep in mind that all results for these abstracts are preliminary because abstracts by their nature include, fairly good data sets, but as the study matures and more data are being accrued and the cleaning, so to speak, of the data is much better and better and better before publication, some of the data will change a little bit. And then most of the studies in T-cell lymphomas, including the abstracts I'm presenting here, are relatively small with less than 50 patients, with some exceptions. So because of that, you really have to take with a grain of salt both the safety data and in particular the efficacy data. And for anything that has to do in terms of, interest and impact and everything else, you're welcome to go back to my disclosures and that presents what my particular interests are. So this is just 4 slides that list all the different abstracts that I selected, which is about 18 or 19 abstracts. Again, I'm not going to present every single one of them, but I wanted to list them here because if once you go back to this video later, or potentially getting the slides, at least you will have kind of the list of all the abstracts that I thought were of interest, starting with the CTCL. And then there are a number of abstracts related to new agents shown here, and I will discuss some of these new agents again. And then some studies about natural history and outcome in biology, which I thought were of interest this year. But again, I'm only going to be able to present one of these studies. But they're all listed here, so you have them. You can go back to it. So starting with cutaneous T-cell lymphoma, the first abstract that I wanted to show is the study from the City of Hope group that looked at the phase 2 trial of a checkpoint inhibitor called durvalumab, which, unlike pembrolizumab and nivolumab, which target PD-1, this actually targets the receptor and therefore has a slightly different safety profile and probably also slightly different efficacy. In combination with lenalidomide, which is an agent that is used as an immunomodulatory drug, the efficacy in a lot of different types of T-cell lymphomas and B-cell lymphomas as well. And so the idea was to kind of put these two drugs together. Not a whole lot of data on durvalumab in T-cell lymphomas, but I thought it was a good idea for this group to run this trial, looking at the primary endpoint being global overall response rate, which in CTCL means you have to look at overall response rate, complete responses in all the four compartments: skin, blood, lymph nodes, and viscera. And the treatment was like this. For the durvalumab cohort, durvalumab as a single agent given on day one, every 28 days. And for the combination, the same schedule for durvalumab given daily with lenalidomide daily for 21 out of 28 days with escalating doses from 10 to 20 milligrams. In terms of the patients were enrolled, 25 were treated on overall, 12 treated with durvalumab, and 13 with a combination. Most patients had a fairly advanced stage CTCL, meaning stage 2B or greater, and 1/3 had large cell transformation. Limiting our presentation here to kind of the bottom line of the study, there was a good global overall response rate with 42% with a single agent durvalumab, which is kind of encouraging. And there was an even better response rate of 75% with the combination. Again, numbers are small here, so we're talking about 12 to 13 patients as a denominator for each cohort, and therefore these numbers are bound to change when we start treating larger number of patients. It was a pretty good median follow-up of 14.5 months. And very importantly, most of the adverse events were grade 1 or 2, with only one grade 4 neutropenia observed. So I think this is a combination of some interest in CTCL, particularly heavily pretreated as this population was. And I'm looking forward to kind of the long-term response data from this study in the publication eventually. Another study which I presented from a much larger clinical trial is called TELOMAK, and this is focused on the health-related quality of life. So the impact of this is the fact that good quality of life data on large data sets in CTCL are not common. There have been previous trials that produced those data sets like the Alcanza and the Maverick. But this is uniquely good because of the number of patients treated, the frequency, the completeness of the data, and because it included a lot of patients with Sézary syndrome, which is an ultra-rare malignancy. So just a couple of words about the drug. Lacutamab is an anti-KIR3DL2 antibody that is in development in a variety of different types of T-cell lymphomas, with the front runner being CTCL and Sézary syndrome. It targets this antigen, which is expressed on a minority of normal T cells and is broadly expressed in high intensity in malignant T cells. The study had initially 3 cohorts, then extended to 4, with the primary cohort 1 being patients with Sézary syndrome that had received at least 2 prior systemic therapies, and the other cohorts being different subgroups of patients with mycosis fungoides based on KIR3DL2 expression in the modality of testing for KIR3DL2. The endpoint was global overall response rate with a number of secondary endpoints, including, importantly, quality of life, which is the data set I presented at ASH. This is the response rates in Sézary syndrome patients. If you look at the first column to the left, the best global response on 56 patients, kind of very encouraging for this heavily pretreated population. So the efficacy is very good. The safety also has already been presented. And here, again, the quality of life data to be presented. Quality of life is a very important issue in CTCL. And we looked at quality of life looking at the pruritus by a visual analog scale, and a number of additional modalities and entities in terms of the endpoints for quality of life, which is called health-related quality of life by Skindex 29. What I'm showing here is a kind of a global view of the Skindex 29 data in Sézary syndrome on the upper right, and then by domain. The way to read this figure is like this. If you look at the bottom and then moving up, each color represent a severity of the quality of life, with orange and yellow representing severe to moderate impairment of quality of life, and green, light and dark green, improvement in quality of life. And essentially, what you need to look is as you go from baseline to longitudinal follow-up. What you want to see is you want to see more and more of each one of those bars being taken by light green or dark green color, and the orange and the yellow colors kind of gradually becoming less and less and less. And that's exactly what we're seeing here in Sézary syndrome, domains being symptoms, functioning, and emotions. And the same is true for mycosis fungoides cohort. And with changes happening fairly quickly. So this was a very good thing to see in this clinical trial with large number of patients and a very dense capturing of data, quality of life data. So the conclusion was that we know that Telemac and there's a robust activity with lacutamab in a favorable safety profile, quality of life focus on pruritus and the Skindex-29. Patients had high pruritus and poor quality of life by Skindex-29 at baseline, and we really saw fairly quickly meaningful improvement from baseline in both endpoints, even starting at week 5 and sustained over time. Importantly, this quality of life improvement happened not just in responders, but also in patients with stable disease. So even if there is stable disease and there's no objective improvement, the patient actually have a better symptomatic disease and quality of life. Now, moving on to some new agents that were presented at ASH. Now, this is an abstract from a multi-site phase 1 clinical trial with a new drug that is targeting CD94, which is a surface marker expressed on a subset of T cells, so CD8 positive, gamma delta, and NK T cell. This trial is important, even though it's a phase 1, it's still relatively small. But it's important because this drug essentially targets a very, very, very high risk subset of T cell lymphomas listed there. Subcutaneous panniculitis, enteropathy associated, monomorphic epitheliotropic intestinal T cell lymphomas, primary cutaneous gamma delta T cell lymphomas, and CD8 positive PTCL. So these lymphomas have very, very bad outcomes, even with frontline therapy. And so there's a huge need for new drugs. And this is the first targeted drug that really attacks those particular lymphomas. So the trial was studied with 2 parts. Part A, which was a dose escalation, and then an expansion in Part B. There were a number of those levels with 3 different induction regimens. And the study started in July of 2022, moving on all the way to May. It's still ongoing. The safety part in the dose escalation was really extremely unremarkable. There was no DLT. The only significant treatment-related adverse event was an infusion reaction, which actually happened in one of the first few patients enrolled in the study. Overall, about a third of the patient had some infusion reaction, which were easily managed. And other side effects were really trivial, with perhaps one single cytokine release syndrome of grade 2. The efficacy, even though, again, this is a phase 1 early stage on 21 patients, but significant responses in multiple different subtypes, including 3 complete responses in very aggressive cytotoxic T cell lymphomas like subcutaneous panniculitis, extranodal NK, and primary cutaneous gamma delta. The response happened fairly quickly. So the average time to response was about 1 month, 1.2 months. And the duration of response for now is variable between a minimum of 2 months to almost 12 months. So very, very encouraging results from this phase 1, very early. But I think that this is something to follow very carefully moving forward. Along the same line, even though this is not a clinical trial, I wanted to point out to the audience here that there was an abstract, which was mostly preclinical, that also was focused on these type of lymphomas. And this is a study from the NIH looking at the development of CAR targeting cells specific for gamma delta T cell receptor. And to make a long story short, they really went from the bench. They designed a variety of different chimeric antigen receptors. They went through the whole selection process. And then they use a number of cell lines listed there to actually do some of the initial in vitro development and some of also the preclinical development in mice using it to treat. The only not criticism, but the only caveat that I have for this study is that all three cell lines here they used were adult ALL, acute lymphoblastic leukemia cell lines, T cells. And that is because there are not many cell lines available from these lymphomas. So they did what they could do. But we don't know that this is going to work for some of the other gamma delta T cell lymphomas. But this is really, really interesting. As you know, just to kind of go over how CAR T are sort of what the structure of CAR T, if you just look at the left here, essentially, the receptor is made of one is called a single chain variable fragment, and then there's a hinge transmembrane domain, and then there's a costimulatory domain. And this is taken from the CAR T cell that we're using for B cells in DLBCL, but the CAR T, the chimeric antigen receptor that they develop for the gamma delta T cell receptor is structured exactly the same way. So we'll see. Another abstract is an interesting study. This was an oral presentation from the Memorial group led by Alison Moskowitz and Steve Horwitz, looking at the combination of ruxolitinib and duvelisib for T-cell lymphoma. So this is a multi-center phase 1 study, and enrolling relapsed refractory patient with PTCL or CTCL, or even very interesting untreated T-cell prolymphocytic leukemia. There was a part 1 for safety, looking at multiple dose levels of ruxolitinib and duvelisib listed there. And then there was an expansion phase for 2 cohorts, patients that had a JAK-STAT2 activation, either by immunohistochemistry or by genetics, and patients who did not, just to see how important the presence of those pathway activation is in these patients. So there were 49 patients enrolled, median age 64, 65, prior therapies, median was 2. And there was a number of follicular helper T-cell lymphomas, about a third of them, another third about peripheral T-cell lymphoma NOS, a number of T-cell prolymphocytic leukemias and other subtypes. And if you look at part 1, there was, looking at all the dose escalations from dose level 1 to 6, the MTD was found fairly quickly for ruxolitinib 20 and duvelisib 25. 25 is the lowest dose of duvelisib, and that was based on some hematologic toxicity seen in the first dose level. Fair enough, that's a good dose combination there. And in the part 2, which was the expansion, there were 23 patients on cohort A and 14 patient cohort B, and the safety there was actually quite good and expected considering these two drugs. The efficacy is interesting, even though again it's preliminary. There was an overall response rate of 41% with 24% complete responses. The median duration of response in responders has not been reached yet. And the signal was quite strong in the follicular helper T-cell lymphomas with an overall response rate almost 80%. And many of them, the majority, were complete responses. Also very, very encouraging here, the overall response rate in T-cell prolymphocytic leukemia, understanding that these are patients who are untreated, newly diagnosed. Although the complete response rate in that group, perhaps not unexpectedly, was 0%. And then there were some responses also in LGL leukemia, which is not expecting. So right now, PTCL and NS, all the other subtypes, not very encouraging responses. So I think that this combination will be explored in follicular helper T-cell lymphomas, perhaps tweaked a little bit in prolymphocytic leukemia, and definitely pursued in T-cell LGL. This is another study, smaller, much smaller, of a combination which I'm presenting it because it's interesting that it's used a combination of an HDAC inhibitor and a drug called tazemetostat, which is not approved in T-cell lymphoma, but it's approved in follicular B-cell lymphoma. Um, the interest in this drug is the fact that there is a similar drug that targets the same sort of epigenetic molecule called EZH1. Tazemetostat only inhibits one of the EZH, and the other drug that is fairly advanced in development in T-cell lymphoma is called valemetostat, and that inhibits both EZH1 and EZH2. So the idea was to see how the combination of these two epigenetic drugs really works. This is a study that was a collaboration between Columbia, Jennifer Amengual, and NIH. Small phase one, standard escalation. The dose of tazemetostat and belinostat are listed there. Tazemetostat is an oral drug. Belinostat, it's an IV drug with a not so very user-friendly schedule because it requires daily infusions for 5 days out of 21 days. The enrollment and number of patients shown here. Overall, I would say a fairly safe combination here with expected profile of safety. There were a number of grade 3 adverse events, and there was one patient who died due to COVID on this cohort. Study drugs were interrupted in 2 and dose reduced in 1, which is not trivial considering that this is just a group of only 11 patients. If you look at the overall response rate, 33% is of interest with some complete responses, particularly in the T-cell lymphoma group. This trial was not limited to T-cell lymphomas. But even 43% is not very exciting, I would say. And therefore, exactly how this combination is going to pan out will have to be assessed. Although I think the idea of combining the two drugs is of interest from a biological standpoint. This abstract, also presented by Alison Moskowitz, is about, Dr. Mehta Shah, Neha Mehta Shah, essentially presented the final results of the PRIMO trial, which is the single-arm trial of duvelisib in relapsed refractory PTCL. This study has been presented many times in the past. This was just kind of the longest follow-up of the expansion phase that included 123 patients. Median age 65, 2 prior lines of therapy, range 1 to 9. Most of the patients had PTCL NOS or angioimmunoblastic T-cell lymphoma. And the median follow-up for the progression-free survival was 6.2 months. This agent, as a single agent, is quite active. I think that's well known at this point. And this drug is available even outside of clinical trials in practice through the NCCN compendium. And in fact, I use it on a regular basis for patients in second or third line because it's oral. And so the response rate is almost 50% with about 33% complete responses. The median progression-free survival is still short, which is, I think, something that is common to all these trials of novel agents in peripheral T-cell lymphomas. The median progression-free survival is always less than 6 months, so a lot of progress there to be made still. But this is a really kind of interesting drug. The issue here, and that's the reason why I'm presenting it, is that the drug, in terms of its approval, is now in a clinical trial called the TERZO trial, which focuses from a regulatory standpoint. In other words, the disease that the company and the sponsors chooses and the investigators choose to actually pursue approval is follicle helper T-cell phenotype T-cell lymphomas, because that was sort of the subset that had some of the best responses in the PRIMO trial. This trial, unfortunately, is only open in Europe and the UK and not in the United States. Part of the reason is that the comparator arm right here, so if you look at the investigational arm, of course, is duvelisib. The comparator is gemcitabine as a single agent, or bendamustine. And these are two drugs that here in the United States are not very frequently used for second line in angioimmunoblastic and follicular helper T cells. So there were some issues about the study design. There were some issues about the regulatory path to approval. And so the sponsor decided to just go and do this in Europe, which I think it's unfortunate. But so we'll see what the results of this study is enrolling. And in fact, at ASH, there was kind of a trial in progress abstract presented by Dr. Zinzani, just describing the trial. Finally, this is in terms of, this is not new agents. I'm sorry about the mislabel there. This is more about outcomes. I wanted to present it because this is the largest sort of retrospective study of T-cell proliferative leukemia ever published. Well, it's not published yet, but ever conducted, and hopefully will be published soon. This is a collaboration between 15 academic centers. This is run by Dr. Jonathan Brammer, who is at Ohio State University. He's the lead PI for this study. A lot of very good collaborating centers. And overall, 377 patients enrolled. The median follow-up 29 months. The diagnosis was confirmed. TCL1 is kind of the specific biomarker for T-cell prolymphocytic leukemia. And this was available not in all patients, was available only in 223 patients, 82% of which did have the TCL1 positive. Interestingly, what the immunophenotype of the T-cell proliferative leukemia, we know that can be variable. In this particular group, 61% had the classical type, so CD4 positive. 11% actually had a CD8 positive T-cell proliferative leukemia, which is less frequent, and then 25% were double positive. So this gives you an idea of what the landscape of the immunophenotype is in a large cohort. A significant number of patients, but almost 50% had lymph node and spleen involvement by PET or CT scan. When retrospectively they look at treatment, they are able to find data on 342 patients, which is 94%, which is a pretty good fraction of the all patient that they had. And looking at any frontline therapy, overall response rate was 67%, with about 50% complete response rates, and kind of median overall survival of 19 months and median progression-free survival of 10 months. So this gives you a pretty good bird's eye view of what the responses and outcomes are with any therapy in T-cell prolymphocytic leukemia. Obviously, a lot of improvement to be made here. This remains a very challenging disease. When we look at the patients who received alemtuzumab as their first line, which is now the standard of care, which were about 250, the response rates were much better. And complete responses, only 58%. I say only because in prior experiences with smaller cohorts, the complete response rate was much higher than that, 80% or so. And so, but the median overall survival and progression-free survival somewhat better, but not strikingly better. And then a small subset was treated with a combination of alemtuzumab and pentostatin, which the authors of this abstract sort of suggested that it seems to be better, but with only 20 patients here, I'm not sure that you can really make a strong statement about that. Besides, the toxicity obviously is going to be significant. Looking at transplant, as many of you know, the goal of therapy for everybody who receives frontline therapy and goes into a complete remission is proceeding to an allogeneic transplant. Here, we see something that we all know, except that is very clear that less than 50% of the patients actually go to of the patient who could potentially go went to allogeneic stem cell transplant. So even though that's the goal, in many cases that's not possible either because of age or because of performance status, comorbidities, access to that therapy and so on. If you do get an allogeneic stem cell transplant, though, the overall 3-year survival is 50%. And if you don't get a transplant, is only 17%. Much improved also the progression-free survival between those who did and did not get the stem cell transplant. So Additional little anecdotes here. Retreatment with alemtuzumab produced a significant number of responses, including 21% complete responses. There's no after alemtuzumab, there's no standard of therapy. People use pentostatin, ruxolitinib, venetoclax, bendamustine, and nelarabine, with understanding that pentostatin is probably the largest group. And there are responses, but not many complete responses. Another observation on this study is that for some reason the CD4 negative proliferative leukemia seem to have a worse overall survival compared to the CD4 positive. We don't quite know why that is. So with this, I'm done. I think we have 15 minutes perhaps for Q&A. I'm very happy to address any question that the audience has. Thank you, Hope.
Thank you so much, Dr. Porcu, for your presentation. And like you said, we're going to get into some questions. So if anyone does have questions, they can submit those through the Q&A box at the bottom of your screen. And yeah, we'll use the rest of the time that we have to take any questions that come in. I did have two questions submitted ahead of time. The first one here is more general. This person is wondering, is lymphoblastic lymphoma a T-cell lymphoma, and how is it classified in the classification system that you showed?
Yeah, that's a great question because it comes up all the time, and I'm not sure that we are very good at sort of explaining that breakdown on the front end. The bottom line is this, that just like in CLL, there is kind of a sister disease or twin disease that is called small lymphocytic lymphoma. So we always talk about CLL and small lymphocytic lymphoma. The same is true for acute lymphoblastic leukemia of T cell lineage. So, ALL comes in B cell, most of them are a B cell phenotype, but there's a subset is T cell. That is an acute leukemia essentially. But T cell ALL has a twin sort of disease that is called T cell lymphoblastic lymphoma. That typically presents with a large mass in the chest in most cases, not always, but and the two diseases are very, very similar, if not exactly the same. So lymphoblastic lymphoma is in fact a T cell malignancy, but is not a mature T cell malignancy, is a precursor T cell malignancy, that falls into the same category as T cell lymphoblastic leukemia.
Thank you. We did get another question in the box. In mycosis fungoides, is CD4 positive mutually exclusive from delta gamma?
Yeah, it's a great question. I would say almost always yes, meaning that from a practical standpoint, the two markers are mutually exclusive. There are anecdotal, bizarre, sort of exceptional situations where there may be kind of a combination of those markers, mostly because these are abnormal cells that, in terms of expressing genes, sometimes they don't follow the rules. But for the most part, CD4 positive T cells, they express a type of T cell receptor that is called alpha beta. And they never or almost never express a T cell receptor called gamma delta. And so the two populations are mutually exclusive in the vast, vast, vast majority of the cases.
Thank you. And the next question here is, what are the side effects of the drugs discussed for mycosis fungoides, and are any of them oral or just infusions?
Yeah, so I think based on the abstract that I presented, I think there were three drugs for mycosis fungoides, which I discussed. One is durvalumab, which is an intravenous drug. It's is a checkpoint inhibitor. The side effect of that drug are somewhat similar to the side effects of nivolumab and pembrolizumab, and they are due to the fact that they activate the immune system very strongly. So you can have some autoimmune manifestations, involving the thyroid, the pituitary gland, the gastrointestinal system, the liver, the lungs, and so on. Less so, I would say. And so that's for durvalumab. We don't quite, there's not a whole lot of information on the toxicity profile of this drug in T-cell lymphoma, though, I have to say. So there's not a whole lot of information. All the information comes from solid tumors. And the other drug was lacutamab, which is the drug used in the large TELEMAC study that is also IV. Um, Unlike durvalumab, which we don't have a lot of information on safety, for the information for lacutamab safety is significant because the trial has completed enrollment, and we have a fairly large data sets on both Sézary and mycosis fungoides. And the safety profile is extremely good for lacutamab. We really know, grade 3 or 4 adverse events, a minimal number of treatment-related toxicities. And so that is an IV drug. Finally, lenalidomide is an oral medication, and lenalidomide has been used in both lymphoma and myeloma now for a very long time, so we have a lot of information on the safety. The primary side effects, it's a pill, depend on the dose, but common side effects include decreased blood count, hemoglobin, white blood cells, sometimes platelets, gastrointestinal side effects, some liver dysfunction, and skin rash. So those are the main side effects of lenalidomide. Very much dose dependent, I would say. Particularly the myelosuppression is dose dependent.
Thank you. Next question here. In nodal PTCL, where there's underlying epigenetic deregulation, do you prefer to use HDAC inhibitors, and why?
Yeah, that's a great question as well. I guess the short answer is yes, just to sort of to cut to the chase. And the reason is that not that necessarily that we have a very good understanding of the mechanism, but just empirically, all the data sets that we have from the HDAC inhibitors being, romidepsin or belinostat or others, and they looking at the subset of patients who had either angioimmunoblastic T-cell lymphoma or one of the follicle helper T-cell expressing peripheral T-cell lymphomas, the efficacy is significantly better with the HDAC inhibitors compared to other drugs. Although now that we're starting to get information on duvelisib and ruxolitinib, those lymphomas also respond quite well to those other agents. I mean, there's no comparison at this point, head-to-head comparison between response rates in these lymphomas with the HDAC inhibitors versus duvelisib or ruxolitinib. But I'm sure at some point those data will come out. HDAC inhibitors are my go-to second-line therapy for patients with angioimmunoblastic T-cell lymphoma. In particular, the follicle helper T-cell PTCLs perhaps are not quite exactly the same as far as response to the HDAC inhibitors, but in the ballpark.
Thank you. The staging at time of diagnosis, i.e. stage 2 for AITL, have an impact on overall survival rates. My understanding is that most participants in studies are stage 3 or 4 when included in the studies.
Yeah, it's true that the participants in most trials are stage 3 or 4. That is because the vast majority of the patients with peripheral T-cell lymphomas, be it angioimmunoblastic or PTCL-NOS, they have stage 3 or 4 disease. It's very uncommon to have limited stage peripheral T-cell lymphoma. Not to say impossible. There's a small subset of patients who do, but the vast majority have advanced stage, and therefore the enrollment on clinical trials kind of reflects the population. I don't think there is a specific effort to exclude patients, except perhaps for stage 1 from clinical trials, but certainly stage 2 to 4 with any type of peripheral T-cell lymphomas are eligible for enrollment on trials. And the prognosis of limited stage 1 peripheral T-cell lymphomas is still a little bit to be defined. In fact, at ASH, there were some abstracts that looked at stage 1, and there are attempts to define risk factors in almost like a prognostic score specifically for limited stage. One actually was even published from the T-Cell Project just 2 or 3 years ago. But it's a very small population. It's difficult to figure out exactly the impact of those prognostic factors.
Thank you. And in mycosis fungoides, with an original diagnosis of large cell transformation, will that continue to be a characteristic of the disease?
Yeah, that's a great question. And the answer is not necessarily. So it's true that mycosis fungoides, the natural history is, continuous relapse or progression over many years. And if somebody has large cell transformation from the beginning, certainly it is, the possibility that the progression at later times will also be with large cell transformation, but not necessarily. For example, if you have like a limited disease site with 1 or 2 or 3 tumors that have large cell transformation, and you treat with systemic therapy and radiation, and those tumors go away, it doesn't necessarily mean that there will be a progression with large cell transformation down the road. That is why, even though large cell transformation is an important prognostic factor in mycosis fungoides, not all patients with large cell transformation actually do poorly. So some patients actually have large cell transformation, the disease goes in remission, and then even though the MF in general comes back, but it doesn't necessarily come back with large cell transformation.
Thank you. Are you aware of any new treatments for relapsed AITL post-autologous stem cell transplant, assuming allogeneic transplant is ruled out due to the age of the patient?
So the answer is about in regard to allogeneic is not necessarily. I mean, I think that there's some patients, it is possible to do an allogeneic transplant after an autologous transplant. Although, as the person on the call rightly says, that if the age is very advanced, the allogeneic doesn't become doable. But allogeneic transplant are now done certainly up to age 65, particularly for fit 65, sometimes even older. So it's possible. It's not impossible. In regard to treatments for relapse angioimmunoblastic after autologous transplant, all the clinical trials of new agents in T-cell lymphoma, including angioimmunoblastic T-cell lymphoma, they're open to patients who progress after autologous stem cell transplant. So there are a lot of patients with autologous stem cell transplant and then go on trial and for new drugs. So there's a lot of options. Yeah.
Your chart showed survival statistics. Were those statistics based upon drug treatment only, or did that include those who also went through a bone marrow transplant?
The survival data that I presented, which are from the Swedish Lymphoma Registry, and there are other sort of data sets that are used, and they're equally good. But that is one that the T-cell lymphoma in the T-cell lymphoma community of investigators we often use because how good the data are, and that includes all patients, regardless of the treatment that the patients get. So that includes long-term survival for patients who, most of those patients were treated with CHOP type regimens on the Swedish Lymphoma Registry. data set, but also include the patients who after CHOP chemotherapy, they had autologous stem cell transplant. So it's really kind of a global snapshot of outcomes, all inclusive.
Thank you. One more question here, unless any other pop in before we conclude, but looks like this participant writes that a transplant has been suggested for them, but they're very afraid, so they're avoiding it. They're wondering if you have any input or suggestions. No more details other than that.
Right. So very tough question to answer without having the context. I guess the first question would be, are we talking about an autologous stem cell transplant or allogeneic stem cell transplant? That would be the big question. I'm assuming that the listener is talking about allogeneic because the risk for autologous is fairly minimal. So I'm assuming allogeneic. If allogeneic, it is always a difficult uh, judgment to make. A lot of times, in situations where I mean, in the front line setting, I'm talking about. So in other words, somebody having one of those very aggressive, refractory, chemo-insensitive T-cell lymphomas, sometimes we make a recommendation to go ahead with an allogeneic transplant, even without patients having failed any chemotherapy, as long as they respond to the first chemotherapy, of course. And so that is called consolidation in CR1, in complete remission, first complete remission. That's a tough call. Usually, the patients who have those lymphomas have adult T-cell leukemia lymphoma is one of those conditions. Patients who have sort of advanced stage the cytotoxic or gamma delta T-cell lymphomas, if they respond, we send them to an allogeneic transplant. So there are very specific situations where, as the doctors in general feel that there is worth the risk, which of course is not trivial, but it is worth the risk because without that, the chances of progressing and dying from progression are very high with those specific types, which is not most of the T-cell lymphomas, but there are some types that are really sort of very poor risk. I don't know if that answered the question, but I know.
I think the best input that you could give is what you
Yeah.
gave. So thank you for speaking kind of generally to that question. We had another specific question pop in. It's okay if you
Mhm.
don't have the answer, but they're wondering if you know the average survival rate for T cell affecting the GI tract. They note that they were stage 2 at diagnosis.
Yeah. I hesitate to give a
Yeah.
number here because, predicting the future is always a difficult thing to do. I would say this, my approach to this is always to some degree personalized. And I know that you cannot draw statistics from individual patients, and sometimes that is a good thing. So, the one, you can look at averages, and for example, the intestinal T-cell lymphomas go from some kind of fairly indolent types, some of those more lymphoproliferative disorders, or the intestinal system or NK cell, those have a very good prognosis. But then the aggressive T-cell lymphomas like enteropathy associated and the monomorphic epitheliotropic, particularly the latter, if you look historically, these are very ultra rare diseases. So again, the outcomes are difficult to really to have some solid numbers, but those are diseases that clearly have a very poor survival on average. And there are some patients who do well and many patients who don't do well. And then just putting a number on a percentage, just based on just a case of an N01 is very difficult. I would be hesitant to do it. And everything depends on, I would say this, I always put this in front of my patients. If you respond well to whatever treatment that the doctor is selecting, that could actually that can put you in a completely different risk group compared to the average, that you extract from the literature and before treatment. So it's one day at a time, one treatment at a time keep keep being hopeful and positive if you see facts on the ground that support being hopeful and positive. And that is a one patient at a time sort of game there. I really feel very strongly that that's the way to approach it with these diseases.
Thank you. I think that's actually an excellent note to end on today. So thank you, Dr. Porcu, and thanks to everyone for joining and submitting questions. Of course, if you have any additional questions after today's program, you can always reach out to our helpline. Their information can be found at all times at lymphoma.org. But for now, their number is 800-500-9976. And the email for that is helpline@lymphoma.org. Again, I encourage you to look at our future programming, especially our Living with Lymphoma program coming up on April 19th, because there will be a T-cell breakout session for those of you who are interested in more information. But thank you so much again, Dr. Porcu. Have a great rest of your day.
My pleasure. Thank you so much, Hope. Take care.
Take care.